Hello everyone, welcome to ITLS Academy. Our lecture is Serology testing and this is part 2. In this part we will study indirect serologic testing. In the part 1 we have covered direct serologic testing. So first of all we will see what are the objectives of this testing. After completing this exercise, the student will be able to perform the following objectives. First one is, in the discussion point 1 is, state the principle and the general procedure behind indirect serologic testing. Point 2, state the difference between a qualitative serological test and a quantitative serological test. Third point, define titer. Point 4, state what disease the RPR and the FDA ABS procedures test for. Indicate which of these is a presumptive test, which is confirming test, and why. Next point, state the significance of non-treponemal antilipid or resin antibodies in serological testing. Next point, state the significance of heterophile antibodies in serological testing. Next point, state the significance of anti-deoxyribonucleoprotein antibodies in serological testing. Next point is, briefly describe the indirect fluorescent antibody technique. Next point, briefly describe the EIA test for HIV antibodies and state the significance of a positive HIV antibody test. Next, state the most common reason for a false negative HIV antibody test. First one is, using antigen antibody reaction in the laboratory to indirectly diagnose disease by detecting antibodies in a person's serum produced against a disease antigen. First point is the concept and general procedure for indirect serological testing. As stated in the introduction to serological testing in previous exercise, indirect serologic testing is the procedure whereby antibodies in a person's serum being made by the individual against an antigen associated with a particular disease are detected using a known antigen. Second point is qualitative and quantitative serologic test. This type of testing is based on the fact that antibodies are only produced in response to a specific antigen. Next point, detection of antigen antibody reactions in the laboratory. Indirect serological test may be qualitative or quantitative. A qualitative test only detects the presence or absence of specific antibodies in the patient's serum and is often used for screening purpose, while antigen antibody reactions may be detected in the laboratory by a variety of techniques. These techniques are agglutination, precipitation, complement fixation, enzyme immunoassay, radioactive binding techniques, and fluorescent antibody technique. Examples of indirect serologic tests to detect antibodies in the patient's serum. First one is the RPR test for syphilis. RPR test means rapid plasma region test, and this test is a presumptive serologic screening test for syphilis. Next is serologic test for infectious mononucleosis. The infectious mononucleosis serological test demonstrated today is a rapid qualitative test for infectious mononucleosis which uses specially treated horse erythrocytes acting as the known antigen that are highly specific for mononucleosis heterophile antibodies. Third is serologic test for systemic lupus erythematosus. Erythematosus. In systemic lupus erythematosus, autoantibodies are made against components of DNA. Next is detecting antibody using the indirect fluorescent antibody technique, the FDA-ABS test for syphilis. The FDA-ABS test is a confirming test for syphilis since it tests especially for antibodies in the patient's serum made in response to the syphilis spirochete treponema pallidum. Fifth one is the EIA and Western blot serological test for antibodies against the human immunodeficiency virus. The Western blot is the test most commonly used as a confirming test if the immunoassay is repeatedly positive. You can see the diagrams. This is FTA-A, this is FTA-B, and this is FTA-C test. So first of all, in the FTA-A, here known antigen treponema pallidum, and these are epitope. In the FTA-B testing, here known antigen treponema pallidum, and these are epitopes and antibodies against these epitopes or this treponema pallidum in patient's serum. In the FTA-C, these antibodies against treponema pallidum in patient's serum are attached to any fluorescent material, and these antibodies fluoresce in the fluorescent light. This diagram shown here indicates immunoassay test. This is immunoassay A, immunoassay B, immunoassay C, and immunoassay D. In immunoassay A, this is known HIV antigens, and this is test well. This shape is test well. In immunoassay B, the antibodies against this known antigen, HIV antigen, are shown here. In immunoassay C, these antibodies are attached over the HIV antigen, anti-HGG antibodies against human body against human antibody are made, and these are attached to this first antibody. In the immunoassay D, color is changed due to presence is this is the enzyme, and this substrate fluorescent substrate for the enzyme attached to the enzyme, and this fluoresce. Just because of the presence of this substrate, there is a color change is observed in the case of immunoassay D. Here comes procedure for the test. First is the RPR card test for syphilis. and we are going to see demonstrations. First point is label 6 test tubes as follows. Ratio 1:1, 1:2, 1:4, 1:8, 1:16 and 1:32. In the next step, using a 1 ml pipette, add 0.5 ml of 0.9% saline solution into tubes, that is 1:2, 1:4, 1:8, 1:16 and 1:32. After addition of saline solution, add 0.5 ml of the patient's serum to the 1:1 tube, and this is undiluted serum. This is the main point of this step. After addition of undiluted serum, add another 0.5 ml of serum to the saline in the 1:2 tube and mix. Remove 0.5 ml from the 1:2 tube and add it to the 1:4 tube and mix. Now again remove 0.5 ml from the 1:4 tube and add to the 1:8 tube and mix. This process will be repeated till the last tube. So again remove 0.5 ml from the 1:8 tube and add to the 1:16 tube and mix. After, remove 0.5 ml from the 1:16 tube and add to the 1:32 tube and mix. And in the last, remove 0.5 ml from the 1:31 ratio 32 tube and discard it. This is not mixed in any of the tube. You can see how 0.5 ml of 0.9% saline is diluted in this test. In the first test tube, there is addition of 9 ml patient serum, and after that in the next tube there is a further addition of 0.5 ml and then 0.5 ml removal from this tube and addition in next tube. From the next tube, a removal of 0.5 ml and addition into the next tube. From the next tube, removal of 0.5 ml and then addition into next tube. And from the next tube, addition into another tube. From the last tube, there is a removal of 0.5 ml and this 0.5 ml is discarded finally. This is discarded. After final dilution, after discard, using the capillary pipettes provide with the kit, add a drop of each serum dilution to separate circles of the RPR card. Spread the serum over the entire inner surface of the circle with the tip of the pipette, using a new pipette for each serum dilution. Remember, there is no second use of the first pipette. Every time for the dilution there is use of a new pipette. Use of same pipette is strictly prohibited. In the next step, using the RPR antigen dispenser, add a drop of known RPR antigen to each circle. Do not let the needle of the dispenser touch the serum. Using disposable stirrers, mix the known RPR antigen with the serum in each circle. In the next step, place the slide on a shaker and rotate for a maximum of 4 minutes. Now read the results as follows. A definite clumping of the charcoal particles is reported as reactive or R, and no clumping is reported as a non-reactive. The greatest serum dilution that produces a reactive result is the titer. For example, if the dilutions turned out as follows, the titer would be reported as 1:4 or 4 dilutions. You can see the reactive are 1:1, 1:2 and 1:4, while the non-reactive titers are 1:8, 1:16 and 1:32. Next, the serologic test for infectious mononucleosis, and this is demonstration. First point, place 1 drop of each of the patient's serum in circle on the test slide. Next, add 1 drop of treated horse erythrocytes, the known antigen, to each circle and mix with disposable applicator sticks. Next point, rock the card gently for 1 minute. Then leave undisturbed for 1 minute and observe for agglutination of the red blood cells. Agglutination indicates the presence of heterophile antibodies. Next is the serologic test for systemic lupus erythematosus, or SLE in short. First point, add 1 drop of each of the patient's serum to separate circles on the test slide. Next point, add 1 drop of the latex deoxyribonucleoprotein reagent that is the known antigen deoxyribonucleoprotein adsorbed to latex particle to each serum sample and mix with the disposable applicator sticks. The next step will be rock the slide gently for 1 minute and observe for agglutination. Agglutination indicates the presence of anti-nuclear antibodies associated with SLE. Next comes the FDA ABS test for syphilis, and this is indirect fluorescent antibody technique. So the point is, observe the 35 millimeter slide of a positive FDA ABS test. Next is the EIA or immunoassay and WV or Western blot test for HIV antibodies. For this test, observe the illustrations of the immunoassay and the Western blot test for antibodies against HIV. This is immunoassay. And this is Western blot. Results. First is RPR card test for syphilis. Detect non-treponemal anti-lipid antibodies and record your results. Next is mono test for infectious mononucleosis. For these results, detect heterophile antibodies. Draw the results of a positive and a negative test. Next is serological test for SLE. First two tests were first one is quantitative, that is RPR test, while the mono test and this SLE test is qualitative test. For the SLE results, this detects anti-deoxyribonucleoprotein antibodies. For its results, draw the results of a positive and a negative test. Next is FDA ABS test for syphilis, and this is confirming test. This detects antibodies against Treponema pallidum. For its results, draw the results of a positive FDA-ABA test or FDA-ABS test. This is not FDA-ABA, this is FDA-ABS test. So thank you very much guys for watching this session. Guys, follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn. You can also subscribe ITLS Academy on YouTube for latest videos and updates, and you can also get it on Google Play Store. Thank you for watching this session. If you like the video, then like, share and subscribe ITLS Academy. For any help or query, you can call us our helpline number 7080-833-450. You can also reach us on www.itlsacademy.com. Thank you very much again for watching this session.